Good morning, everyone, and welcome to today's MPG Primer. We are very excited to have three speakers with us here today to speak about cell painting and how image information can illuminate disease variants. Our three speakers today will be in this order. The first is Dr. Ann Carpenter. She's an institute scientist and director of the imaging platform at Broad, where she focuses on extracting useful information from images using computational tools. Our second speaker will be Juan Caicedo, who is a Schmidt Fellow at the Broad, who specializes in machine learning for biology. And then finally, our third speaker will be Dr. Samira Asgari, who is an assistant professor at the Institute for Genomic Health in the Icahn School of Medicine at Mount Sinai. Her work focuses on identifying variants, underlying complex traits, and understanding their function. We are very excited to have all three speakers here this morning who will be speaking one after the other, and they're happy to take your questions during the talk. So please feel free to type questions in the Q&A or raise your hand and be acknowledged to voice your questions. So thank you very much, and Dr. Carpenter, lead us off.
Great, thank you. Welcome, everybody. I wanted to point out this link here, although I think I haven't actually uploaded the PDF of the slides to this folder yet, but if you want to make note, feel free to take a screenshot of this chunk of it, and we'll have that posted later on today. Welcome. I'm excited to be talking about cell painting today and describing work from actually a lot of collaborations with a lot of different Brodies and beyond. And we'll be doing this tag team style so that you get a sampling of a deep dive into a couple of questions. So I'm going to start pretty high level, pretty broad, mostly focusing on the conceptual, because I know for many of you, using images as data is not the most familiar thing in the world. So let's get started. I first want to begin with, oh, and thanks, Scott, for putting that link into the chat. I wanted to begin by telling you about just the basic concept of image-based profiling. We use the cell painting assay, but image-based profiling refers to this broader strategy of taking images, all their beautiful pixels, whatever channels you have available, extracting the features from them at a single cell level, and you get this gigantic, beautiful data matrix that's just like a transcriptional profile or a proteomic profile or metabolomic profile, but in this case, we're capturing morphological features of each individual cell. That gives us tons of data, which we can do all kinds of profiling applications with, including clustering samples based on their similarity, looking for differences in samples, such as a disease variant versus its reference counterpart, and so on. So we'll talk about a lot of examples of what you can do with it, but the basic concept is that we're taking these lovely pictures, microscopy pictures of cells or organisms or MRI scans of brains or whatever it is that you can think of, and extracting their features and then using those features as data to look for similarities and differences among patients or among samples. So that's a basic idea of image-based profiling. And when we use the cell painting assay, a typical experiment would look something like this, where we choose either a single cancer cultured cell line, or we use primary cells, or we take patient cell lines. Somehow we take some sort of cells, we add some sort of perturbation, drugs or genetic perturbations optionally, and then we do these high throughput experiments in 384 well plate format, where we stain these cells, carry out image analysis and feature extraction usually using classical image analysis, such as what you find in Cell Profiler, but also sometimes we're using now deep learning to extract features. And then this gives us this type of data, which we can then do downstream analysis with. And here's what the assay itself looks like. It's not particularly critical to understand it, but the basic concept is that we're staining organelles that are very broad and very general. They give us a general sense of the structure, the different structures that are within the cell, and they're broad enough to capture a lot of different kinds of perturbations, as you will see. So nothing very specific to a particular pathway or to a particular biological process of interest. They're really quite general. And a side effect is that these are very, very cheap dyes. They're not antibodies, so you can carry out this assay very inexpensively, less than $2 a sample. So what can we do with that? That's all fine and good, but how much information is really in images? And I wouldn't blame you if you're a little bit skeptical that just snapping a picture of a cell tells you anything about what that cell is experiencing. But we were very excited in one of our early experiments to look at genetic perturbations where we were overexpressing a couple hundred genes. we did the cell painting assay, we did the image analysis, and then we clustered those images based on how similar the cells look in each image using this numerical data. And we were thrilled to find that 50% of the genes in this set gave a phenotypic signature that was distinguishable from negative controls. So here's about 100 of them on this plot, because the 100 that didn't have much of a phenotype are not shown. And to a cell biologist, this is just a beautiful thing to see, that this one experiment using only image-based data is able to recapitulate a lot of the different pathways that have been taken decades for a biologist to painstakingly deconvolute. So we've got BRAF, KRAS is up here, we've got the hippo pathway down here, we've got the NF-kappa B pathway and other groupings that make sense. If you have isoforms of the same gene, they tend to fall nearby each other. If you have a constitutively active version of a protein, it's nearby the wild type, but not quite on top of it, and so on. So it's really exciting to see that a lot of common sense biology made sense. And we even found that not only do proteins in the same pathway tend to cluster together, but if you have a negative and a positive regulator of a pathway, often they'll have an opposite phenotype. And that's one that we discovered in this experiment here. All right, so I just want to take one little brief digression. I talked about the cell painting assay being very generic, and this experiment showed us it can cover a lot of biology. It's not just a teeny fraction of the biology that's captured with these 6 stains that we use. But sometimes people are studying areas of biology that are much more, are interested in interrogating morphology that's more specific. And so I won't be talking about it today, but in the fall, Melina Klausnitzer's lab will be giving a talk about lipocyte profiler, where instead of using kind of a standard cancer cell line, U2OS, which is our common cell line for cell painting, they instead use primary human adipocytes, and then they're also adjusting the stains so that they've added the BODIP to label the fat globules, and they find all kinds of interesting things, and I'll leave it to them to talk about this fall. So one can take this general strategy that I'm describing and make it much more tailored to a particular biology if you wish. But we do find that we get pretty far with just the generic cell painting, and most of the projects I'll talk about today stick with the with the standard cell painting. All right, so let's say that you've profiled a lot of genes, you have a particular signature, a particular morphology that's induced by your gene of interest. Wouldn't it be nice if you could just immediately translate that into drugs, into compounds that are able to either mimic the activity of your gene or oppose the activity of your gene? And it's a little bit of a long shot kind of project, but we gave it a try anyway, where basically we're carrying out a virtual screen. So we take of those 100 or so genes that you saw on the prior slide, we tested 69 of them, where we just, in a completely virtual experiment, we just took the signature of a given gene from that experiment, and then we tried to match it against 15,000 compound signatures that we had available from an experiment almost a decade ago now. And long story short, it did not work all the time, as one could expect. If this worked all the time, then all the pharma companies could stop doing what they're doing and go very quickly find drugs for everything. But we were pretty impressed that it actually worked 19% of the time on a set of known gene compound pairs. So for particular compounds that were known to target particular genes, we tested this relatively small set and found that 12 of them gave the correct compound at towards the top of the list. And so we were pretty excited by this. It might not sound like all that much to say, 19%, I wish it was 100%, but may not sound like that much. But if you tell a pharma company that they can reduce their screening costs by 19%, you can see how happy they are, because this is something that they, of course, spend millions to billions on collectively. So I won't go into any of the details, but it's one thing to prove this retrospectively on compounds and genes that we already know match, and there's kind of ways to overfit things, and maybe you don't trust it quite so much until we discover something new. So then we went to discover something new, and this was a beautiful story of a lot of Brodies helping out, where we took the predicted compounds for 7 genes and just handed them out to all of our friends who studied different genes. So it was really wonderful to have people helping out here. And for 3 out of those 7 genes, we found chemical inhibitors of the gene that they were interested in. A couple of them, some of them had not great stories, like they did regulate the gene of interest, but in the wrong direction for therapeutic purposes. And in one case, that they were relatively weak inhibitors of the compound. But nevertheless, the basic strategy worked. And in one of them, we actually did find some good pathway regulators. So that's a cool thing that you can translate from a gene signature into a drug signature and potentially find good drugs. So because this is such an exciting direction, because this kind of thing is working so well, we've been working with 10 different pharma companies and another nonprofit to create the world's largest public cell painting data set. So it will have, the details are shown here, it will have a ton of small molecule signatures, as well as overexpressed genes and CRISPR knockdowns. Still nowhere near the level of transcriptional data that's in the world. I mean, there's millions of transcriptional profiles, but we're trying to get to the point where there's a big enough data set that people can do the kinds of queries that I just described in a virtual way by matching. So let me take a step back. If you've tuned out, you could tune back in now, because I'm going to change a little bit of a topic here and talk about, okay, one way to get a signature of interest is to perturb the gene, or maybe overexpress your gene of interest and see what it does to cells. And so if you've done genome sequencing, you have a list of genes, you don't know what they do, you very much might be excited about grouping those genes into pathways to try to figure out the function of some of them if it's otherwise unknown. You could find a drug that targets it, like I just described. But another way to come at this is to look at the variant level. And Juan is going to give a much deeper dive into this sort of a concept, but I want to give one example here where if we take this particular protein and tag it, it looks like this in whatever cell type, HeLa cells. And if we take that same protein and give it the mutation that is known to cause a human disorder called Noonan syndrome, the localization of that protein changes. So very simple experiment where you just overexpress the wild type version or the variant, and you see a difference. And once you see a difference, it doesn't happen all the time, but if you do see a difference, you can test for existing drugs to reverse that phenotype. Or you could test genetic perturbations to figure out what proteins and genes are involved in the pathway going from healthy to pathogenic. So that's the basic concept of this project that we collaborated with Mikko Taipale's lab at University of Toronto. He actually tested 3,000 of these different variants with their wild type reference counterparts, and then identified changes, not just in the protein's localization. You can see some of the more obviously visual examples are shown here. Some are much more subtle, but there were also a few other channels in the assay, not exactly cell painting, but a few other organelle channels. So we can look for morphology changes there as well. And in the end, we have a few hundred disease phenotypes that are discovered from this project. And you can imagine if you've just completed a genome sequencing experiment, what if you could take all the hundreds of variants that you've uncovered, maybe some are below the threshold of significance, maybe some are above it, but why not just test all hundreds of them and see what the impact is on the protein's localization? Maybe the answer is nothing detectable, but in some cases you may find some kind of change in localization that can give you a clue as to the function of that gene and the impact of that variant, and also just allows you to group variants, as Juan will describe in more detail later. So the really exciting thing is we're going to be doing 80,000 of these variants in an IGVF consortium project. There's 2 IGVFs happening at the Broad, 2 different centers. This particular one is testing 80,000 coding variants, and that work is just getting going. So as I said, we're kind of at this point, and when you get to 200 disease phenotypes, we would like to do a drug screen for each of these, so for every situation, take the disease looking cells, add 2,000 or 3,000 FDA approved drugs and see if we can find any that cause the protein localization to go back to normal, which may or may not cause the function to go back to normal. But it's really, it's not so bad doing a few chemical screens at this scale, but we can't really do 200 of them. So what can we do about that? And the answer is we can pool the reagents in a technique that was first developed by Paul Blaney's lab and then adapted by JT Neal's lab. What we can do is we can take literally 24 well plates, so not super high throughput, but in each well, we're not just testing one compound for one disorder, we're actually testing each compound, already approved drugs, against 200 reference genes and the 200 disease variants that I mentioned in the previous slide. So in each case, these pairs have a different localization, and for each drug, we ask, does this drug help any of the unhealthy looking cells go back to the healthy looking localization or morphology? And it's a very cool technique. The basic strategy of how this works is to infect the cells with the pooled sets of variants, treat each well with a different compound, then image the localization and morphology first, and that's what that looks like, and then remove those fluorescent stains and do a cyclical in situ barcoding technique to detect which cell had which protein and which one had which version, the variant or the reference sequence. So that'll be underway. That concludes what I wanted to share with you today, but I think perhaps the most important thing is to be aware that there's a lot of ways that you can be involved. I love presenting about this research because it's not one of those talks that you come away with thinking, oh, that's really cool, but I can't do anything about it. There's lots of ways that you can make use of this sort of data in your research. You can profile genes and assign them to function based on similarity to other genes with known function to see if they match other genes. You can see if they match other compounds. You can profile sets of alleles, as Juan is going to dive deeper into, to figure out which alleles are impactful, which effects are similar to each other. You can identify disease phenotypes in images. This is a very small scale kind of experiment to do. These require that you make sets of reagents in kind of a larger scale project of at least a few hundred samples. For identifying disease phenotypes, you literally just take, patient cells of two different sorts and look at them first, perhaps by eye, even if not computationally, or you can take larger groups and do a more intensive computational analysis. Once you see a disease phenotype, or once you detect one, you can then see if those phenotypes can match genes or compounds and look for reversal. And then lastly, if you have sets of compounds, I guess that's probably less common in the crowd of MPG, you can see, again, if they match genes, other compounds, and see whether there's specific morphological phenotypes that are induced by them. So these are some of the ways you can get involved. And in particular, I really wanted to emphasize that right this moment, the IGVF consortium is gathering up genes and alleles where they will carry out the cell painting of the reference and variant versions of proteins. And they will also be doing protein-protein interaction profiling in yeast of the same genes sets. So if you're interested in getting any of your genes or alleles into that consortium, it's happening, and they are doing 80,000, and they're happy to take suggestions for which ones to add into that pipeline. So it's a really excellent opportunity to get this kind of thing done for free with essentially no commitment on your part. So with that, I will conclude my section, and I'm happy to answer questions about it before we turn to Juan.
All right. So this was a very nice introduction by Anne about how you can use morphological profiling for studying diversity of problems in drug discovery and functional genomics. It's my great pleasure to present this other project in which we also used cell painting for variant impact phenotyping. And this project was run jointly with a team of scientists, spanning people from the cancer program, the imaging platform, and some other collaborators. So it's my great pleasure to present this for you today. All of the data and the images and the preprint associated to this work are available in this URL. In case you are interested in learning more about this project, feel free to find it there. So the motivation for this project is that we want to understand and be able to characterize whether variants that are reported across many different diseases can be identified as impactful or non-impactful in using some sort of systematic assay. And as you probably know, there is a growing number of variants of unknown significance being reported every year. These statistics are from before the pandemic. The number of variants that are identified in sequencing samples is growing exponentially. And this is something that we would like to understand better in order to design treatments. So in our project, we were looking specifically at cancer variants, and more specifically, lung cancer. So we used the cell painting assay in order to create a data set where we tested a pool of variants that were already reported as being present in the tumors or samples taken from real patients in the clinic. The set of variants was tested and introduced in a subset of A549 cells. This was a high throughput experiment using overexpression. And for each of the variants, we used 8 replicates. In total, the dataset has more than 5 million single cells that we were analyzing in order to find differences between the reference genes, which are also known as wild types, and the variants that we are studying in this project. So here are some example cells taken from the wild type samples for some of the genes included in this study. The first thing that we did with this large data set was to transform the images into quantitative data that we can analyze. So this pretty much follows the same pipeline that Anne described before, including image analysis, normalization of the data, and finally creating the profiles for each of the variants or wild types that we have. This is an example of the control cells in one of the plates. And the controls in this case are wells without any overexpression. So we call them empty controls. This is a reference gene, BRAF in this case, and this is one of the variants included for this specific gene. We looked at the replicability of the multiple replicates that we had. Is it consistent? And what we found after this analysis is that the morphological features are consistent across replicates. So more than 80% of the treatments that we use for this analysis find correct replicates in a matching experiment or a matching simulation. So the quality of this data is very high. So next, we move to try to model and address the question of how different are the variants that we have in this reference set with respect to the wild types. And for that, well, we also observed kind of the overall set of genes in this study. And as you can see, this is the list of the 50 or so genes that were included. For each of those, we had about 8 to 20 variants included. This is the matrix of similarity across the replicates. And one thing that we observed is that the variants of each of the wild type genes that we included kind of cluster all together. So for instance, all of the BRAF variants that we have are similar to each other here, and same happens with KRAS and other genes. There is a lot of them that come together, kind of replicating what Anne also mentioned before, that genes that belong to the same pathway are also related morphologically. And this was also observed in this data. We also looked at the clusterings from another perspective, in this case using UMAP plots. And again, if you see, one of the wild type genes, which is in green, you're likely to observe the variants hanging around this in the morphological space. So what we want to do now is to understand what is the effect of those variants with respect to the reference gene. And we used the variant impact phenotyping algorithm that was proposed for this type of analysis a few years back by Alice Berger and others at the cancer program. In their case, they were using this algorithm to understand the effect of variants or the impact of variants using transcriptional profiling. So we are kind of testing the same ideas here with morphological profiling. And basically, the basic idea here is that we have a distribution of correlations or similarities between replicates of the reference gene and replicates of the variant for that specific gene. We can measure the cross-correlation between the morphological samples in the reference gene and the variant, and we can look at those distributions and interpret them in a statistical way. So using decision tree, we can identify whether the variant has a gain of function mutation, a loss of function mutation, or a change of function mutation as well, using the same structure as it was used for transcriptional profiling. Here are a few examples of the 3 or 4 different types of outcomes that you can get out of this analysis. For instance, this loss of function mutation for KIEP1 indicates that the reference gene has a very high and strong correlation across all of the replicates. And when you look at the variant of this specific gene, which is this one, you see a drop in the correlation of the samples, which indicates that the phenotype of this variant is doing something different, first of all, with respect to the reference gene. And given that the phenotype is weaker, we also infer using the decision tree that this is a loss of function mutation. We can see gain of function mutations when the reference gene doesn't have a strong phenotype, but then the morphology of the variant becomes more different or stronger than the original phenotype of the reference. And that indicates a gain of function mutation as well. We ran this for all of the variants in our set of cell painting data. And what we can observe here is that some of the genes, for instance, STK11, has most of the variants have a loss of function mutation in this case. And the way we interpret these plots is that we have the negative log p-value of the impact in the x-axis. So larger in this case is better or more impactful. And in the y-axis, we have the directionality of the prediction, whether it's a loss of function mutation going down in the y-axis or a gain of function mutation, which goes up in the y-axis. So these ones are gain of function mutations versus these ones, which are loss of function mutations. And we can kind of visually see what are the set of mutations that we have in each of the genes. We can also look at single cell resolution in this data set because imaging allows us to look at the differences in morphology between sets of single cells. And in this case, we are comparing again a couple of variants from the BRAF mutation. Each point in this UMAP plot is one cell taken from the replicates of this specific overexpression experiment. And we have 3 populations depicted here. The orange one is cells from the variant treatment. The reference gene is in green, and control cells, which are empty controls, are in blue. So what we can see is that single cells in the phenotypic space are moving to different regions of the space, depending on what variant you're looking at. So in this case, the W450L variant is moving towards this state of the phenotypic space, whereas the V600E is moving to another place. Both of them were classified as gain of function mutations in the VIP algorithm that I described before. But when you look at the single cell resolution, you can see that they are kind of gaining different functions, if you like. And this would be interesting to look at in detail. We also measure the correlation of the signal that you get with cell painting with respect to transcriptional profiling. For a subset of the genes and variants used for this study, we also had L1000 data from the previous study run at the cancer program. So we looked at how much signal we get for the variants using the cell painting assay in this y-axis of the plot. And in the x-axis, we have the counterpart for L1000, how much signal you get for each of those variants or wild type genes. And what we see is that there is a high correlation between them. So if you have high signal for that specific variant in L1000, it's likely that you're going to observe also a high signal phenotype using the cell painting assay. We also used canonical correlation analysis to see how well aligned both assays are with respect to the correlations across multiple variants. And in this joint latent space, we can see a variance in green indicating the gene expression profile and variance in blue indicating the morphology profile. Red lines are connecting a couple of replicates across the platforms, which indicate that they are relatively well aligned in terms of a phenotypic signal. We also looked at the predictions of transcriptional profiling using a subset of common variants between both platforms. And we observed also kind of a lot of agreement in terms of the number of impactful variants observed by L1000 with respect to the ones observed by cell painting. I think they agree a lot and they are highly correlated, but there are also very interesting differences between them, which we also explore in a little bit in our preprint, because even though the assays and the platforms have a lot in common, they also complement each other, and they are telling us different things about the variance in this case. In conclusion, cell morphology is very useful for variant impact phenotyping. And we also observed that image-based profiling allows us to explore single cell heterogeneity. And overall, cell painting predictions correlated well with L1000 predictions for this problem of variant impact phenotyping. So I think that's all I have in my presentation. If you have any questions, let me know. And thank you to our collaborators and all the people that contributed to this project.
Thank you so much, Juan, for an amazing talk. There are two questions I would like to voice, and I will just do that right now. The first one is, how do you convert the images into a numerical vector representing the phenotypic readout? And would applying different methods to do this conversion lead to different readouts, potentially causing the variants to cluster differently?
That's a great question. I think we ran this specific analysis with different sets of features. And we started with features obtained with the Cell Profiler software, which is the software that the imaging platform has developed. And you can get features that are the so-called manually engineered features. So you can measure, for instance, the area of the cell or the size of the cellular compartments and get all of those numbers as a description of the morphology for every single cell. And then you aggregate that information across replicates. There are also newer methodologies for extracting features based on deep learning. So in those cases, instead of defining the features ahead of time, you let an optimization algorithm find what are the most relevant features for this specific set of images that we have. I think running both, we did a comparison and we pretty much obtained a very similar answer. There are differences. Sometimes the type of features that you use allow you to see certain differences, and in some cases you don't see the differences. So the quality of the features matters, and we are investigating on how to optimize the type of information that you can extract from the images.
Great. Thank you very much. And the second question is one that I think is relevant to both Anne's talk and yours. Here it is. The audience member wonders whether the alleles are most of the time, as you're referring to here, coding variants, and whether the process is fundamentally different in some way between coding as compared to assessing coding as compared to non-coding variants.
Yeah, I think that is also kind of a great question. I think for this specific study, the selection of the variants was recommended and suggested by the team of experts in the cancer platform. I think it was a very similar process as Anne asked before, like, contacting collaborators and asking which variants do you guys think that would be very interesting to see in this project. I believe that some of them were, I think it's in the preprint, but I forgot off the top of my head. But they are, yeah, sorry, I think I forgot, and I was going to ask Anne, but she dropped from the call as well. If you want more information, maybe we should, chat online and clarify more, because I feel like I cannot answer this right now. Sorry about that.
No worries at all. Thank you. And it looks like Anne may be on the way back online. So we will check in with her again later, I hope to.
Okay,
Thank you so much, Juan, for a wonderful talk.
Thank you.
Okay, so I hope everybody is hearing me. If it's otherwise, let me know. And today, in continuation of the two great talks that Anne and Juan presented about cell painting and some of its usage, I'm going to talk about the genetic basis of cell morphological traits. But before starting to go to cell morphology's genetics, I just want all of us to be on the same page. So let's talk for a minute about the genetic basis of human traits in general. As you probably know, human genetic traits or diseases can be put into a spectrum that ranges from monogenic diseases to common complex traits. And although there is no clear cut between these two, the general belief in the field is that the monogenic diseases or Mendelian diseases are caused by one or just a handful of variants. These variants tend to be protein coding and tend to be rarer in frequency in the population. And as we move from monogenic traits to more common complex traits, the causal variants for those traits become, first of all, more frequent at population level. And then their effect sizes tend to be smaller, and also they tend to be, they are more likely to be in non-protein coding regions of the genome. So as you can imagine, most of the genome is not protein coding, and as a result of that, most of the variant disease associations that we have fall on the non-coding part of the genome. So just to give you an example, here is the pie chart showing the proportion of variants associated with autoimmune diseases through the study of common complex autoimmune diseases, and about 90% of these variants that has been identified do not affect the sequence of the protein in the genome, and about 70% of them do not even fall in a gene. So they are in non-gene region, intergenic enhancers, regulatory regions, but not in the gene. And the issue that this causes is even when a variant is in a gene, and even when a variant does change the protein sequence, it's not always easy to know how it affects disease outcome. This challenge becomes much more complicated and difficult when the variant is not even in a gene. So a major problem right now in the field of human genomics is to understand the function of non-coding variants, but the function of disease-associated variants in general, but especially non-coding variants, and then understand how this relates to disease outcome. So over the past few years, there has been a number of efforts in the field, because as of a few years ago, the field as a whole realized that we've spent over a decade identifying genetic associations, a large part of it through genome-wide association studies. And now we have thousands of variants that are associated with a wide range of traits, from non-disease traits like height to autoimmune and lots of other diseases. We have this large pool of variants, but for most of them, we cannot really bring them to clinic because we do not know the variant function, and we do not know how that function relates to disease outcome. So over the past few years, this effort has started in the field of human genomics and complex trait genetics, that usually is referred to as V2F, short for variant to function, or M2M maps to mechanism to medicine. But basically, regardless of how you call these efforts, the goal is to connect genetic variants to function to clinical outcome. And there has been a wide array of methods that has been proposed and used to achieve this goal. Here, I'm just showing basically a summary of all these different methods. The point is not to go through them one by one, but just to show them that the methods that has been proposed and used range from purely statistical fine mapping methods that try to find the causal variance in the genetic region to much more complex methods such as working on genetically modified animals. In all this range of different methods, something that falls kind of in between these two sides of the range in terms of effort and time are what we call quantitative trait or QTL analysis. And for those of you who may not know what a quantitative trait locus is, it is a locus, it is a specific place in the genome that changes, basically that variation in that locus is associated with change in a quantitative trait. That quantitative trait may be gene expression, it may be chromatin markers, it may be cell morphology. The figure that I show here, the scheme that I show here shows an EQTL, an expression quantitative trait, where a genetic variant of a specific locus affects the expression of a nearby gene. And over these past few years, when people started doing more and more QTL analysis, something became clear very early on that QTLs are usually highly context specific, meaning that a given variant may be a QTL in a specific type of cell, but not a QTL in another type of cell. The context may be disease, maybe tissue, maybe a specific point in time, but the general belief now in the field is that most QTLs are context specific, and this is something important to take into account. So this brings me to cell morphology. A lot of different QTL analysis has been done over the past few years. Most commonly known are expression quantitative trait loci. However, cell morphology is one that is less explored, and I think Anne and Juan's talk showed that it is a good one to look at as a cellular trait for the purpose of QTL analysis. There are good evidence to tell us that cell morphology has a genetic basis, so it can be linked to genetic variants. Famous example is from sickle cell anemia, where a mutation, it's a Mendelian disease, but the mutation in the gene basically changed the shape of the red blood cells. But there are other examples of more common variations in the population that basically affect the shape, the volume, the hemoglobin capacity of different blood cells. But the majority of these previous studies have focused on blood cells. The major reason for that being blood is an easily accessible tissue, and the morphology of blood cells are well known compared to many other cells in the human body. So there are good scientific reasons to look at the genetic basis of cell morphology. There are also another good incentive to take cell morphology as a basis for QTL analysis and for understanding the function of genetic variants, and that is cost. Juan mentioned in his talk that we can do cell morphology profiling at single cell level, and I just told you that cell context matters a lot for many QTL analysis. But one big bottleneck in using single cell level data in many other types of cell profiling methods is the cost. For example, here I just listed the cost of RNA profiling, chromatin profiling, and single cell morphology profiling. And as you can see, there is a huge difference in terms of cost, whether you do it in bulk or whether you do it at single cell level. Whereas for cell morphology, because it's an imaging based, basically you do look at single cells anyway, and then it's up to you how to analyze the data, but the cost is even for bulk much lower than other cell profiling methods, and then at single cell your cost does not increase. So all these are really good reasons to use cell morphology as a quantitative trait to basically perform QTL analysis. And this is exactly what we did in collaboration with a number of colleagues at the Broad Institute and the technology that Anne talked about. Basically, this slide is to give you a general idea of the big picture of what we've done, we started by about 300 cell lines. So cell lines obtained from 300 different individuals. These were induced pluripotent stem cells. I'm going to come to them in a second. After plating and imaging, we end up with a matrix of cell features for each cell. And I'm not going to go to that because I talked about it. But at the same time, besides the imaging profiles that we generated from the cells, each individual was also sequenced. So we also have whole genome sequencing data from each of these 300 individuals. And then what we can do is to basically look at the association between genetic variants and some morphological features. Before going to any results, just a side note on why we decided to go for induced pluripotent stem cells and not other cell types. You probably know these are cells that have the capacity to become differentiated to many types of other cells in the body. And I told you that context matters a lot for QTL analysis. So the idea here was that we start by these iPSCs, and for any variant association that we may find, we can then differentiate these cells to many different cell types and check whether that association is still active in the specific context of a different cell type. And then clinically speaking, iPSCs are valuable because the idea is because they are from a patient, after differentiation, they can potentially be transplanted back to the same patient, basically shortcutting a lot of potential immune rejection issues. The 300 cell types, the 300 individuals that we started from were about 50% male, 50% female. Some of them were coming from individuals with specific diseases, and then they had a range of ages and a range of different ancestries. In the next few slides, I want to show you just my goal here is not to go to the details of what we did in terms of QC. What I'm trying to tell you to give you a sense is the extent of the data, where we started, how we dealt with this data type that basically was completely novel because it's not every day that people generate such in-depth cell profile, cell morphology profiling data. So we start by about 4,000 features and over 5 million cells, which is a very, very large number of cells if you're familiar with single cell technology. And then we started basically at the first step, removing some features and removing some cells based on a number of criteria that are listed on the slide that I'm not going to go to. But this first QC step basically left us with about 3,000 features, and most of the cells passed this QC. So only about 3% of the cells were removed. After this initial QC, we had about 3,000 features, but something that I mentioned in her talk was that many of cell morphological features are highly correlated. So for example, if you have a bigger cell, so if your cell area is bigger, it's very likely that your other feature, that is cytoplasm area, is also a larger number. There are lots of these types of correlation between your 3,000 or so features. And when you're doing statistical tests, having a large number of correlated traits or features can cause you a very high penalty in terms of multiple testing. So we decided to reduce this number by selecting a set of quote unquote uncorrelated features. But when I say uncorrelated, some of them are still highly correlated. We just, after a number of benchmarking, decided to put that threshold at Pearson correlation coefficient of less than 0.9. And doing this from 3,000 features, we suddenly could reduce our feature space to about 250 features. And then we have the variants from whole genome sequencing. Not all 300 individuals had whole genome sequencing. About 297 of them had the sequencing information. And we started from about 40 million variants. And after running a set of standard QC steps, we ended up with about 7 million variants that are common at the population level. By common, I mean they have a frequency over 5%. And now we were ready to go to the next step, which was basically finding the association between genetic variants and cell morphology features. This is our QTL analysis, what we call CMQTL or cell morphology QTL analysis. And in the simplest model, you can imagine that we just regress a given feature on a given genotype at a specific locus. And this could work, but it's also rational to think that besides genotype, there are many other factors that may affect cell morphology. For example, differences in sex, differences in age may affect also cell morphology. And when you do this kind of modeling, it's always important to remove the effect of those non-genetic factors to be able to find true genetic associations. So one obvious thing for us to correct for was differences in genetic ancestry, just because differences in genetic ancestry can affect differences in allele frequency or presence or absence of the alleles in the population. So that was the first step. We knew that our samples come from different populations. We can perform, using their genetic data, we can perform this analysis called principal component analysis and basically take that differences in genetic ancestry into account in our model. This was something obvious to do, but then the question was, what else can we correct for that can potentially affect our results, but are not necessarily directly related to the effect of genetic on cell morphology? And we had a list of potential confounders to choose from. They are listed here. And when you end up with 10, 12 more number of potential covariates for your model, the question is how to choose from them. One simple way would be, let's correct for everything. Let's keep all of this in our model. That's not generally a good idea. First of all, because you can overcorrect for things and reduce your statistical power by including too many covariates. And then some of these covariates are highly correlated. And again, by including them all in your model, you can reduce your power to detect true associations. The way around this usually is to do something called variant component analysis. And when you do this, basically you ask the question that of all the different potential covariates that I have, which ones explain a significant amount of variance in my desired feature or in my desired trait. And basically you go and for each feature covariate pair, you ask that question, how much variance in this given feature is explained by that given covariate. And what you see in these plots are basically the result of that variant component analysis. And without going to the details, basically we ended up with 3 covariates that seem to be to have a large effect on the majority of the features. Those were the number of cells that each cell was touching, and the average cell count per plate. So not just the cells that are immediate neighbors of the cells, but how densely populated the specific well in a plate is. And then finally, which plate, so batch effect, which batch basically that imaging result comes from. And after correcting for these plus population structure, so basically differences in genetic ancestry that I talked about, when we correct for these factors, we see that any remaining covariates does not explain a large amount of variance in our data. So here we were all set and ready just to go and test our associations, which basically is shown, the results are shown here. This is the Manhattan plot of all the tests that we have done, which shows basically the association of 7 million variants with 246 features. So each dot represents the result of one of these tests. The total number of dots in this plot is 7 million times 246. What you see here, the red line is the red, because we've done such a large number of tests, we need to correct for multiple tests. So the red line that you see here is basically the statistical threshold for the significance of results. Anything above this line means that it's a statistically significant result. And as you see, there is only one place, small locus here on chromosome 22 that passed this statistical significance threshold. As you may imagine, initially we got really excited about this result, and we wanted to know what variant exactly is associated with which feature in this locus. And we went and looked at it, and the name of the feature may not tell you, or honestly does not tell me much, but the point was there is a variant associated with something that has to do with nucleus shape. But in genomic association studies, every time you find an association, the first thing that I learned to do, and I think anybody should do, is when you get a positive results and your excitement is over, ask yourself, is it a true association, or how can I prove that this is not a true association. And then if you can not prove yourself wrong, you can get excited again. So that's exactly what we went and did. And Juan mentioned that cell morphology features are highly correlated. So it's rational to think if my variant is associated with a given feature, it should also be associated to the same level of strength or similar level of strength with other features that are highly correlated with that given feature. And this is what basically this plot shows. The odd thing here was there was a feature that was highly correlated with our nuclear shape feature, but the p-value for the association of our variant with this feature was really low. This was the first point that we started suspecting these results, and after much digging around, we found out why this association is being, what this association is being driven by. And basically the answer was, there were among all our features, there were 8 that besides all the covariates that we have corrected for, they also were associated with sex. And this specific feature related to nuclear shape was one of them. And then after we corrected for sex, basically in our model, the association that we saw went away. So this is before correction for sex, when you see this kind of classical QTL plot. And after correction for sex, you see that the association goes away. And the reason for the sex association was that this specific locus in the genome is one of the ones that has a very biased kind of allelic heterozygosity based on sex. So most of the reference alleles, homozygous reference alleles, comes from females, and most males have homozygous alternate allele or heterozygous allele. So what did we learn? So I started this talk telling you that cell morphology has a genetic basis, and I ended it by telling you that we did not find a significant association. What does that mean? Should we stop doing this kind of analysis? Based on our experience and based on what we learned through this project, the answer is no, definitely not. What I believe is happening here is that we just don't have enough statistical power. There are good evidence that I didn't have time to go to from our data that there is actually a genetic basis to cell morphology. We just need probably bigger sample sizes or maybe more powered methods to find them. And then the conclusion here is that when you start dealing with new types of data, there are a lot of non-trivial quality control steps that you may need to deal with, and cell morphology is one of those new types of data that compared to, let's say, RNA sequencing, when you started, there is a standard pipeline to go through. This is not the case. And all those steps are very informative about learning how to deal with your data. So if any of you in the future wanted to deal with this type of data, we are more than happy to help. And that's basically our story up to here. I would like to thank everybody who helped with this project. And I would just like to take 10 seconds to say that I just started my lab at New York City in Mount Sinai Hospital. And we are just beside Central Park with great views on the park. And I'm looking for postdocs, so ping me if you're interested.